Please start. Okay. That was my hint. Welcome everyone to the Institute for Advanced Study at City University of Hong Kong. It is my distinct pleasure to welcome all of you to today's talk by Professor Michael Yang. Unfortunately, this has to be all online, not hybrid, not in person, and we all know why. Thanks a lot for joining us in this venue. Before I give the floor to Professor Yang, let me briefly introduce him to you. Professor Michael Yang is the Vice President, Research and Technology, and Yung Kin Man Chair Professor of Biomedical Sciences at our university, City University of Hong Kong. Michael received his Bachelor of Science from Xiamen University and the PhD from the University of Toronto. He then moved on for postdoctoral training at the Scripps Research Institute in California. Was also affiliated with the University of California, San Diego. As he will speak today, his research focuses on studying cancer biology and developing novel technology for diagnostics, therapeutics, but also research applications. Michael has been extremely productive in his career, and since joining CityU in 1994, so a few moons ago, he's published more than 300 peer-reviewed papers, has more than 30 patents, trained countless PhD students, over 30 postdocs in his lab. His age index is impressive and stands at 52 in Scopus. Not only is he productive in terms of publication, he also hoards prizes. He's been awarded the K.C. Wong Foundation Award in 2003, the Chinwe Scholar Award by the Ministry of Education in 2004, the Shenzhen Science and Technology Innovation Award in 2006, the Hong Kong Technological Achievement Award in 2007, the Natural Science Award by the Ministry of Education in 2015, and the Wuxi AppTech Life Science and Chemistry Award in 2016. He also holds honorary professorship at Zhejiang University, the Army's Medical University, and the Institute of Microsystems of the Chinese Academy of Sciences. He currently also serves in various government agencies here in Hong Kong, including the Research Grants Council, the Innovation and Technology Fund, the Health and Medical Research Fund, and the Hong Kong Genome Institute. I would be remiss if I didn't mention his founding or being involved in the founding of two very successful companies. One that we all know all too well in the pandemic, Prenetics and Salomics, that are based on technology that were developed in his lab and at this university. So Michael, it's really a pleasure to welcome you to this talk. It's as we realized earlier, it's very popular, and without any further ado, the floor is yours.
Thank you. Thank you, Klaus, for the very kind introduction. Klaus is my dean. He's the dean of the Jockey Club College of Veterinary Medicine and Life Sciences. And so first, I'd like to thank the Hong Kong Institute for Advanced Study to invite me here to give this distinguished lecture, the first one in 2022. And it is a challenging time in Hong Kong. We have gone through a couple of years of pandemics, and right now we are experiencing a major outbreak here, the 5th wave of Omicron. And so I thank everybody who joined the lecture today, either from your office or from your home. And I believe that with the joint effort by everybody, the community, the Hong Kong AR government, with the strong support from the central government, we will overcome and we will look forward to getting back to our normal, hopefully soon. Now, before I start my lecture, I want to first give 1 minute of a commercial for the Hong Kong Institute of Advanced Study. Hong Kong Institute of Advanced Study is established by City University of Hong Kong as the flagship research institute that assembles all the leading scholars here from many different disciplines to promote free thinking, free exchange, and explore the frontiers of science and engineering and all other disciplines. So I'm honored to have the chance to give a lecture similar to the other well-known scholars from outside and within CTU, including our president, Wei Guo, who gave the lecture a couple years, I think last year, and also Klaus is one of them. So we have regular distinguished lecture series. And in addition, we also feature a rising star lecture, cover different areas from mathematics to physics, chemistry, and life sciences. And that introduced the upstarts, the young and promising scholars from the university. And the Institute also organizes a number of symposiums a year, again, cover the interdisciplinary areas of science and engineering. So I would encourage everybody to go to the website, and there's always new information and new activities that would be of interest to you. Now today, I'm going to share with you some of my research group's recent research over the last few years. We've been focusing on developing new technology and apply to study cancer biology, and hopefully can leverage on the discovery to develop new diagnostic and therapeutic for the cancer. So today I'm going to talk about our research on single-cell analysis of tumor heterogeneity during cancer metastasis. Now, as Klaus mentioned in his introduction, my research group's interest is on the development of new technology, including biochip and nanotechnology, to study cancer biology and to develop innovative molecular diagnostic and therapeutic applications. So these slides summarize some of our research interest and our publications over the last 5 years. And this is also accumulated in this book that I edited and also contributed. So this will highlight our research efforts in developing of detection methods for precision medicine. Now, for today's topic, we're focusing on cancer metastasis. Now, cancer, we know, is a major challenge to human health. And according to World Health Organization, there's a subunit under WHO called Global Cancer Observatory, and an estimated 19.3 million new cancer cases and 10 million cancer deaths occurred worldwide in 2020. So this is the alarming figure showing that our public health and our individual health are under threat. Now, if we look at the figures in China, it's really alarming. 24% of newly diagnosed cases and 30% of cancer-related deaths are coming from China in 2020. And China's age standardized incidence rate and also the mortality rate are way above the global average. And as you can see in this bar chart, comparing the cancer incidence and deaths between 2015 to 2020, and we can see there's a continuous increase across all type of cancers. Now, the primary cause of death, more than 90% of them, is because of cancer metastasis. Cancer metastasis is a very complicated physiological and cell biological process. As you can see in this diagram, which I copied it from these excellent review published last year, the primary tumor undergoes metastasis. It is a cascade involving 5 key steps, including the invasion, intravasation, the cancer cells moving out of the primary tumor into tissue and invade and pass through the circulation system, and then find a niche to extravasate and then colonize to develop into a secondary tumor. So this is the common understanding now about the metastatic cascade. But the detailed mechanisms, the molecular and cellular process involved are still not well understood. And it is important that we know exactly what happened at the molecular and cellular level so that we can find appropriate markers that can predict the occurrence of the metastasis, and then we can develop novel therapeutics that can inhibit or stop the process. Now, the metastatic niche, if we further zoom in to the tumor cells, the activation of invasion and metastasis by a tumor cell involving a number of genetic and epigenetic factors, including the environmental stimuli, biological and physical chemical signals. There's a chemokine, cytokine, other growth factors. The extracellular matrix exert a number of mechanical, physical strengths, and also a number of epigenetic factors, including circadian rhythm and aging. Circadian rhythm means the timing. If you don't sleep or you spend overnight too often, that's going to cause some epigenetic changes that will lead to the cells to undergo different behaviors, including invasion and metastasis. So this is a very complicated system, and we describe this complicated system using the word called heterogeneity. Tumor is not a single cancer cell grow, multiply. The tumor itself is a very complicated system involving cancer cells, cancer-associated fibroblasts, infiltrated immune cells, and a number of other things. Even the cancer cells within the tumor are different because of their different clonal lineage, and they are tumor-initiating or cancer stem cells and other different phenotypes. And the tumor situated in the microenvironment, that including the surrounding blood vessel, the extracellular matrix, and all other biological and chemical signaling molecules and circulation, so on. So the whole system is heterogeneous in terms of genetics, phenotypic, functional, and also microenvironmental. So the research challenge here is the in vivo modeling and study of tumor heterogeneity and microenvironment, because we need to do this research in a lab instead of inside the human body. So the in vitro study is very important. So in the next half hour or so, I'm going to use a number of examples, a number of projects that has been carried out in my lab over the last few years. Some have been completed and published. Some are still ongoing to illustrate how we are combining the engineering approach from the technology point of view, developing various technology, primarily using microfluidics, and in combination with some single-cell analytical tools, including sequencing and atomic force measurements, to study the metastatic processes. Now, the first example is the study on the cell migration, because the first step of the metastasis is the cell migration, moving out of primary tumors. So the cell migration is also under a number of different factors, affected by a number of different factors, including chemical and physical factors. So here we are talking about a cancer cell will be subject to different chemical molecules in the surrounding, and also to subject different physical. But one example is the electrical field around this tissue level. Okay, so we want to see how we can model these systems in vitro using microfluidic devices, and then to study how cancer cells migrate under different conditions. So this is one example we developed to study the cancer cell chemotaxis. So the movement of cancer cells under a chemical attractant. Now the chemical molecules inside the circulating system, inside the tissue, is not homogeneous. The concentration exhibit in the gradients. So we need to generate a device that can control the gradient of these molecules. At the meantime, allow the cancer cells to move in these gradients. So this is one example. We developed a microfluidic chip with the gradient control and also the channels that allow for single cells to move. And this provides a quantitative model to study the chemotaxis at single cell level, and we monitor the migration rate, the cytoskeleton and morphological change, the phenotypic change, and also the metabolism of the cells under this gradient. Gradient means the concentration can be going from low to high or high to low. Okay? And we discovered that the cancer cell chemotaxis actually is exhibit a gradient-dependent migration for morphology change, and also the metabolism alteration, and involving specific signaling pathways. Another example is the electrotaxis, the migration of cancer cells under a small electrical field gradient. So again, our body, there are some electric potential. There are electric fields in our different tissue, different organs. And this is important, for example, for the wound healing. Once you have this information, there's also create different electrical field strengths around the tissue site. And cancer cell migration also occur in this electrical field. So again, a few years ago, we have published a number of papers on the development of microfluidic chips that can generate electrical field gradient in the microchannels where single cancer cells can migrate. And we use that to study the cell heterogeneity, because even, as I mentioned, for the cells from the same tumor, they are different. And even for the cells from the same cell line, they are also different. So we observed the heterogeneity of these cells in terms of their migration potential, and we are also able to isolate these cells depending on their migration rate, and then further study their molecular mechanism underneath. So these are some of the work that has been published already. And another factors affecting these cell movement is the mechanical stress. And we know tumors are is a solid mass situating in the complicated and matrix with different stiffness. And the cancer cells, when they invade and extravasate, they need to squeeze out of the tumor mass and through the gap of the tissue cells into the circulating system. So during the process, they are subject to a various degree of mechanical stress pressures. So how does this mechanical stress affecting the cell migration? So this is something that we want to look at. So we developed these microfluidic chips that comprise of single cell migration channels with different dimension, such that cells might move in the open free space or in a space that has a confined dimension where cells have to squeeze through. So interestingly, we find out that cancer cells actually prefer or actually migrate faster in the space, in the confined space, than in the open space. So as you can see here, so we have these channels with different dimension in terms of the height, 6 micron to 30 micron. And the cells, the volume, the diameter of cells about 15 to 20 micron. So when they are moving in the channels with 6 micron, they have to squeeze and lay flat. And we see that actually they move faster along that way. Interesting, when we look at the cells migrating in these channels, and we measure their elasticity, their stiffness, using atomic force microscope, and we find out that the cells moving faster in a channel, what we call the leader cells, exhibit a much higher, much bigger elasticity, or a less stiffness than the follower cells. So here I introduce another term called cancer cell plasticity. Now plasticity actually in cancer cells has two meanings. One is on the mechanical side. Mechanical plasticity means how elastic they are. And biological plasticity means the phenotypic change. Cells have different phenotypes depending on their internal epigenetic and depending on the proteins that they express on the surface. So as the cells moving along these stress environment, and we also find out they undergo phenotypic change. And these phenotypic change are associated with these mechanical changes, plus elastic changes. Okay? So the migration rate is in the confined channel is associated with cell elasticity, and which is associated with a phenotypic change called EMT. Now what is EMT? EMT means epithelial to mesenchymal transition. Tumor cells are of epithelial origin, but when they are going through the process of metastasis, it is known that will undergo the epithelial to mesenchymal transition to make it phenotype that are more invasive. So the cells, cancer cell from a tumor can decimate it, can move out of the tumor as a lone wolf, as a single cell, or as a group. So what we call the single cell dissemination or collective migration. So there are some questions that remain to be answered. For the single cell dissemination, the question is, do these cells move out by randomly, or they have some predetermined factors? And when they move out, the previous understanding is that the epithelial cells move out and gradually undergo the EMT change into a mesenchymal phenotype. So our question is, do these cells undergo EMT before or after? Now for the collective migration, on the other hand, a group of cancer cells move out together. Are they the same or different? So what's the heterogeneity among these migration groups? And they move out not marching in the same rate. So there are some cells move in the front and some follow in the back. So what is the difference in terms of the plasticity between these leader cells and follower cells? So those are the questions that we ask. And then we start to design some technology to study these, try to answer this question. So here is another microfluidic device that we developed where we have single cell migration channel. We can trap single cancer cells and then look at how they move under different conditions in the confined space or in the open space. And we can also measure these single cells using the atomic force microscope to look at their mechanical plasticity and stain these cells to look at their phenotypic plasticity. So we are doing these single cell profiling, looking at their EMT-related genes expression. So the result shows cells with more elasticity enter the confined channels and migrate faster under mechanical stress. Now, migrate faster, we already find that out. Now here, when we have the cells at the entrance of the channel, they are distributed randomly. So we don't know which cells are more stiff or less stiff. But after they move into the channels, when we measure them, they generally become softer compared to the cells randomly distributed in terms of their Young's modulus, which is the mechanical elasticity. In the unconfined space and confined space, there's a significant difference in terms of the elasticity. And so we also look at the gene expression of these single cells, particularly the genes related to the EMT. And we find out there's a significant upregulation of the EMT-related genes among those cells that are migrating into the channels. So the answer we've derived from these is cells with greater elasticity in the beginning, because the cells has a distribution because of the heterogeneity. They are preferred to migrate into confined space. And the EMT occur before. They are partial EMT before cell move in and continue to undergo further transition during the migration process. So that's about the lone wolf, the single cancer cells. What about the group of cells? When the group of cells are moving, they move like a finger. So there's a tip of cells leading a bunch of following cells moving out through the gap. So similarly, we want to answer this question. We want to perform a profiling of these fingers, of every cell within the finger, to see what kind of elasticity profiling and transcriptional behavior related to EMT genes. So this is, again, we developed these devices whether we can form these cancer fingers during the migration, and we are able to, using a single cell picking device, to pick the individual cell and measure using qPCR the expression of EMT-related genes. And at the meantime, we can also use the AFM to measure the elasticity of each of these individual cells within these fingers. So again, what we see is very similar. The leader cells at the tip exhibit a much greater elasticity. That means they are soft, flexible, compared to the follower cells. So this is the number of cells that we measure from the beginning, 1, 2, 3, 4, 5, 7. And you can see this increase in terms of the stiffness in the cancer cells. And also, we measure the transcription profile of these leader cells compared to the cells way in the back and identify the upregulated genes. And among the different type of cellular model, we find out there are a number of genes and signaling pathways that involve some of the common ones, including vimentin and SNEL, two of the important EMT-related genes are significantly upregulated at the fingertip, as we can see from the in situ immunostaining, as well as this is the mapping of the gene expression based on qPCR result. So we can see significant upregulation of SNEL and vimentin, and when we inhibit these two genes, and the migration front becomes very blunt, the fingers disappear. So this shows that the heterogeneity among the migrating cells are different. They are spatially dependent transcription and stiffness profile among these cells. And we see a continuous increase of phenotypic plasticity and mechanical plasticity from follower to leader cells. So these are the few examples that we have completed, looking at the cell migration from these under these different chemical, physical, and mechanical factors. Now, those are using the in vitro and well-established cell lines to study. So we'll start looking at what about in the clinical samples, in a real cancer situation, what happened. So this is one project that we carry out a couple of years ago, looking at the single-cell transcription profile of epithelial ovarian cancer ascites. Ovarian cancer ascites are the cells, including single cells or cell clusters, that disseminated from the primary tumor in ovarian cancer into the peritoneal cavity. And these are the ones that can undergo a number of processes and generate secondary tumors. So, the presence of these ascites correlate with poor prognosis of ovarian cancer. So the question is, what are the EMT transcription status of these ascites single cells and cell clusters? Can they be classified based on the EMT expression profile? And for those cell clusters, what are the cells inside these clusters? Because the cells inside the clusters ranging from tens of cells to hundreds of cells. Are they the same, or how are they different? So again, we developed a protocol to isolate the ascites single cell and clusters and analyze them using the transcription profiling. So from the different clinical samples, we isolated dozens of single cells and cell clusters And for the cell clusters, we also further disperse them into single cells and then carry out the EMT transcription profiling for every of these cells, and then carry out some cluster analysis. And we observe they are, from the different pathological definition of the ovarian cancer, they are intrapathological similarity and interpathological differences at the molecular level, which is expected. And we also identified that they are intra-cluster and inter-cluster heterogeneity. So different clusters, they exhibit different composition of different type of cells. And within the same cluster is not coming from the same type of cell also. Okay, so these are the different composition. We find out that they are primarily three types of cells: epithelial-like cells, and cancer-associated stromal cells, and some immune cells. And they are different among the different population. And when we're further looking into these profiles, and we find out that actually the similarity between the single cells from the individual cell and the cells from the cluster, the cancer-associated stromal cell, they are similar from different patients, different clinical samples. On the other hand, the tumor cells, they are quite different. And it is known that the tumor clusters are more metastatic than the individual cells in the ovarian cancer. Now, why is that? Now, because of the cluster itself comprised of different elements. They support each other. For example, the tumor cells within the cluster, they have upregulating of EMT-related genes, which exhibit higher migration and invasion abilities. On the other hand, the associated stromal cells, they have upregulation of ECM-related genes, which enable stronger cell-cell interaction and prevent anoikis. So the EMT activation is related to chemoresistance and DNA damage repair. So these provide us with understanding in terms of the chemoresistance, and may provide some targets to improve the chemotherapy and radiotherapy. Now, this is talking about the disseminated cell. If we're looking back into the primary tumor and the metastatic tumor, then we want to see, can we find something in common? Because the secondary tumor is always coming from the cells from the primary tumor. But can we identify these initiating tumors in the primary site? So this is the study, another study we carry out related to ovarian cancer. Now this study, in addition to looking at the EMT only, we look at the whole transcriptome using the single-cell RNA-seq technique. So we have collected a number of tumor samples. Some are primary tumor, some are metastatic tumor, which means they are diagnosed with peritoneal metastasis before chemotherapy, and also some relapse tumor. They are recurrence of tumor several years after surgery and chemotherapy. So we want to find out whether we are able to identify the initiation population of tumor cells in the primary tumor from starting single cells in the relapse and metastatic tumors. And we also want to find out the role of stromal cells in the tumor microenvironment. Remember, when we see the ascites, the stromal cells play an important role in the metastasis. And hopefully that we can identify some biomarkers, some genetic molecular markers for prognosis and prediction of recurrence. So we're using these single cell technology to analyze the samples. And these 8 different clinical samples, altogether we have collected more than 10,000 tumor cells and 3,000 stromal cells and carry out the transcriptome sequencing, RNA-seq. And very interesting, we find out that tumor cells are different from different patients. So the 8 different patients, the tumor cells are clearly distinguished into 8 unique, distinct clusters. On the other hand, the cancer-associated stromal cells, the microenvironment, they are similar. They mixed up together. We have fibroblasts, we have macrophage, we have ovary and other cytotoxic T cell, endothelial cell. You see the 8 different kind of, coming from 8 different samples, they mix together. So this is very interesting. So this tells us that the cells are clustered according to their tumor origin. Fibroblasts are the dominant cell population in the microenvironment. And metastatic tumors contain more immune cells and less tumor cells than the other tumor sites based on the analysis of the percentage of these different types of cell types. And we're further using different bioinformatic analysis to look at the origin of these cellular population. There are tools based on the RNA splicing and also RNA expression called RNA velocity analysis or pseudo-time analysis that can trace a population of cells based on their transcription profile to find out on the time scale what are the earliest cell population among these group of cells. So using 2 different bioinformatic approach, we are actually able to identify a group of cells in the relapsed tumor that have similar transcription profile. And these cells can be mapped into the primary tumors. That means in the primary tumor, they also contain a group of cells that exhibit the similar property as those cells that happens the earliest in the relapsed tumors. So these give us a hint. So we further look at these cells and find out that the subpopulation of, in addition to the initiating tumor cell, there's also a subpopulation of cancer-associated fibroblasts that contribute to tumor metastasis. So as I mentioned, there are 2 populations of fibroblasts. One of them are metastatic in nature. The other one is a primary tumor, do not involve in the metastasis. So we also identify the genes that are upregulated in these particular population. And using these genes as biomarkers, we are able to measure from the tissue side the significance, the correlation between the expression of these markers. So they are markers from the cancer cell and markers from stromal cell. When we combine them together, they provide a good prognosis indication for the outcome, for the relapse-free survival of EOC patients. So that shows the power of these single cell analysis. Now we further move on. As I said, when the cells are disseminated from the primary tumor, they will move into the circulating system. So these cells or cell clusters are so-called circulating tumor cells. Now these are extremely rare. In the cancer patient, the number of circulating tumor from 1 milliliter of blood are in the number of maybe a few to a few dozen. Now, in 1 milliliter blood, we are talking about 10 million white blood cells and other cells, and 100 million red blood cells. So in order to isolate these few cancer cells from these 100 million cells, it requires very sensitive technology. And over the years, the research community has been developing different kind of technology for the CTC enrichment and isolation, ranging from using physical methods to biochemical methods, using positive selection, which try to get the needles from the haystack, or negative selection by removing the other type of cells and then try to get what's left. So I'm not going to get into detail. Again, there's a very good review published 10 years ago. And what we have done, okay, the slides, okay, slides moving. Okay, so again, we spend some effort to develop a microfluidic platform for CTC enrichment and isolation and characterization. Now, cancer cell is different from the other cells. So by using the difference in terms of their size, in terms of their plasticity, mechanical plasticity, and in terms of the surface protein, we are able to combine these different features and develop a microfluidic system that can separate these very few cancer cells from the majority of the other cells in the blood. So by combining the size-based separation with the epithelial and mesenchymal markers, we are able to capture and isolate these circulating tumor cells. The reason we're using these microfluidic approaches is that we want to isolate these cells alive without damaging them, so that we can carry out subsequent culturing and analysis. So this is the platform that we have validated using almost several hundreds of clinical samples. We collaborate with a dozen or so of hospitals, mainly in mainland China. And we can see that CTCs can be detected in majority of these tumors. We can see that with CTC count of 1 and above, we see that more than 97 on average, ranging from 85% to 100%, different type of tumor. And the threshold, because in the healthy control, occasionally we also see some of these CTC or more or sort of a rare circulating cells. Now, the number of these CTCs are also associated with the stage. Obviously, this is understandable, expected, because the later stage will have more cells disseminated into the circulating system. So using this device in combination with the single-cell sequencing technology, we want to get the insight into the molecular signature of these CTCs. So we, using the whole genome single-cell sequencing approach. Now, previous study was using single-cell RNA-seq that's looking at the expression profile. For RNA molecule, when the genes are transcribed, there are thousands or even more of copies of RNA. So in terms of detection, sensitivity-wise, the genome sequencing, there's only one copy for each cell. Okay, so we have established a protocol where we can isolate these CTCs, already very rare, and then we are able to amplify the DNA from the single copy of genome and to carry out the sequencing. So our task is try to find out, is the genetic signature of the CTC identical or similar to the primary tumor? And whether there's any different CTC-specific mutations that may be related to the metastatic potential. And whether we can find out there's some CTC-specific markers or targets that can be used for diagnostic or therapeutic intervention. So this is the study we carry out on a number of, this is work on a liver cancer. So we have collected almost more than 100 of liver cancer patient blood samples, and then to analyze the CTC status. And we find out that the CTC number is associated with the tumor size, is associated with the degree of macrovessel invasion, and also majority of these CTCs are mesenchymal in nature. The epithelial CTC is only 17.5%. And the whole genome sequencing result, we analyzed hundreds of these single CTCs, reveal that the concordance between the genome of primary tumor and the genome of these CTCs are in the range about 90 to 96%. So they are closely resemble that of the primary tumor, but they also carry some of the unique features that may confer their metastatic potential. And further analysis, again, these are all unpublished results, further analysis revealed that some of these somatic mutations are associated with tumor stages. For example, the mutation spectrum are related to vascular invasion, to distal metastasis, and to the tumor size. So our conclusion based on these studies that the mutated genes in CTC are highly consistent with those in primary liver cancer, suggesting that CTC can be used to reveal genomic features of tumor and also to identify the drug targets when the regular tissue biopsy are not available. And the whole genome sequencing of CTC also provide us with a lot of information with regard to the mechanism of the metastasis. So this we still continue to work on along this line. Now finally, I want to share with you one of the latest work that we have been embarking on is to go to the third generation of the sequencing. Now, previously, when we talk about single-cell sequencing, it involved either a group of single cells disseminated from the tumor, dispersed, so we lost their three-dimensional information. We lost their spatial resolution. And the CTC itself, of course, is the isolated cells. So a recent technology that has been named the method of the year by Nature in 2020 is called spatially resolved transcriptomics. That means you can carry out single-cell RNA-seq in situ in the tumor sample without disrupting the three-dimensional or the tumor structure. So this technology is very powerful. It's a combination of a on-chip hybridization and single-cell RNA-seq. So basically, the tissue section from the tumor can be first stained to generate its pathological image. And then further, on the specialized design chip, where the chip contains of thousands of spots, each spot coded with millions of these barcodes. And these are unique barcodes for that particular spot. So when the tissue section are sitting on this chip, then every cells on that tissue section are coded with a unique barcode. And then these cells will be then subject to the single cell RNA-seq, and the barcode information can be used to reconstitute the whole tissue image of the RNA expression. So this is a very preliminary result we just obtained a while ago. We have carried out these spatially resolved transcriptomics of liver tumor sections. We have two cases. We obtained the tumor sample after surgery, and we take a section from the middle, from the center of the tumor, and also from the margin, from the frontier of this tumor. And we want to compare the cells at the frontier and the cells in the center. What are the differences? Because the ones at the frontier exhibit the metastatic related properties. So these are the tissue sections and the RNA-seq results that we obtained. So we can see that for each of these, there's about 3,000 to 4,000 of these single cell RNA-seq results of these sections. And then this can be these single cells can be, again, subject to the same clustering analysis. You see these 4 different sections, the cells can all be grouped together and then do the cluster. That will identify these different cell types based on their transcription program. And then these different subtypes can be using the unique barcode to map to the tissue original pathological image. So what we have seen from these 2 samples alone, we see that the samples actually comprise of 11 or more clusters. And the clusters are specially mapped to the tissue. Then we find out that for the tumors in the center, compared to the tumors at the margin, the ones in the margin are more heterogeneous. We further differentiate these different cell types in the tumor center margins. So, as you can see, for the tumors in the center and the tumors in the margin from the same patient, the profile, the composition is quite different. And for example, in the tumors at the margin, we see a predominant cell type here, the cluster 2, while it's not seen only a very small population in the center of the tumor. Similarly, for the second case, we see a predominant cluster in the margin, cluster 5 here, and again, only a very minor component in the middle, in the center. So these are the cells of interest, because these are the ones that we're seeing that play some roles in the metastasis. So we can map these cluster cells into the tissue section, and indeed, they all expose around the margin of the tumors. Now this is overlaid with the pathological H&E imaging. And you can, if you further look at the percentage, you can see that the ones for the sample number 1, we have predominantly this cluster 2. And for the sample number 2, this is predominantly sample cluster number 5, which is only occupy a small percentage in the center of the tumors. So these are our preliminary results, and we can further look at the genes that are upregulated in these cell types. Just from the 2 patients alone, we compared the upregulated genes in cluster 2 and 5 individually, and find out that actually there are a number of genes and pathways that are shared among these 2 samples. And these tells us that these are the genes that may be the main driver for the cancer cells to undergo metastasis. And these, we have looked at these genes and identify a few that are related to cell metabolism and ribosome biogenesis, and we are continue to look into the downstream mechanisms. So, my time's always almost up. So, I've shared with you some of our recent research work, and you can see that this is a continuous effort in understanding the fundamental biology of cancer, particularly cancer migration involved in the cancer metastasis cascade. And we're also using the technology that we developed along the way and the discoveries that we made along the way to develop novel diagnostic or therapeutic approaches to better control and fight the cancer. And this is the last slide to show that this war on cancer is ongoing. And as I showed in the very beginning, cancer poses a significant threat to the humanity, and we need to work together. And as I show some photos here, in both China and the US, 2 of the largest countries in the world, at least we share a common goal here, to fight and win the war on cancer together. So, for example, the precision medicine initiative in the US and China's precision medicine program launched a few years ago. The primary goal is try to develop new diagnostic and therapeutic approaches against cancer. And recently, the US government launched this cancer moonshot, try to improve the survival rate of cancer patients and try to eliminate the cancer in the foreseeable future. And similarly, in China, the central government launched this Health China 健康中国2030 program. So this, again, aiming at significantly reduce the cancer incidence rate and mortality rate among the population. So I'm very confident, despite the complexity, and I'm very confident that with the efforts by the medical scientific community, we are slowly, but we are winning the war on cancer together. So with that, I want to thank the funding agencies that provide the support for my research over the years. Specifically, I want to thank Dr. Yong King Man, a philanthropist in Hong Kong. They provide support to my research. And the Tong Foundation. This is the foundation by Tong's family, our former chief executive, Tong Chi-Hua's family. They make a very generous donation to support the biomedical research in CTU and my research group. And these are my collaborators. I have a long list, and these are some of the key collaborators that involved in the projects that I mentioned during the lecture. And finally, I want to thank my students, my former students and current students, for their hard work and their dedication to research. I'm very grateful to have the opportunity working with all of them. So thank you very much for listening to coming to the lecture today. Thank you. I'll be happy to answer any questions.
Thank you very much, Michael, for this very impressive talk and technologies, and not only the technologies, but applying them to real-world problems of an important issue, which we all know cancer is. Thank you very much again, and I'm happy to entertain your questions from the audience. Please either type them in the chat forum or raise your hand so that your questions may be read and answered. So before everybody gets going, I don't see a hand up yet. Michael, it was one of your first slides was very interesting, showing the cancer incidence in China being higher than in other parts of the world. Is there any explanation, sensible explanation for this observation?
Yes. There are a number of factors involved. There are some socio-economic factors. China has the improvement in the economic status, so there's a change of lifestyle that are involving the diet and other lifestyle change. The aging population, we are entering into an aging population in China. The baby boom in the 50s and 60s right now are entering into the late senior years. There are some environmental factors, the environmental pollution that is widespread for the past 40 years. So all these contributed to the increase in the cancer incidence. And so, as I mentioned, the central government actually in their Health China 2030 has set up the goal to try to reduce this cancer incidence and mortality rate.
Chufang, I see your hand. Please ask your question. Please don't wave at me or Michael. Please use the hand in the Zoom function or the chat, because there's too many participants to really oversee all the waving hands. I just happened to see Chufang's hand. Please go ahead, Chufang. You're still on mute, Chufang. Oh, I need to unmute you. Ask to unmute. I am.
Okay. Yeah. Thank you.
Okay.
Thank you. Now I can. So Michael.
Yes. Hi, Chufang.
Yeah. So I would like to know that in your in vitro system, so you see the cancer cell migrate, and you say there are different environment. How that could apply in the in vivo system?
Now, I mean, again, I mean, this is an in vitro model because it's difficult. I mean, it's impossible in the past, but now there are some technology that may be able to track cell movements in vivo, but still very difficult. So what we are trying to do, I mean, is taking this reductionist approach, try to eliminate all factors except one, and try to develop some kind of a simple but quantitative model system. And I believe that information or the knowledge that we generated through these models, eventually they can be integrated and apply and simulate what happened in situ, in vivo.
Now, the next question is, in the in vitro system, definitely we don't have the immune system there. Can we not also apply the immune system, some CAR-T, all those technology, and to see how to simulate in a real situation?
Yes, I think this is, again, another area of active research in the area of this technology, microfluidics and in vitro cancer research. People are developing different ways of growth, tumor, spheroids, or organoids, and to mimic the situation inside by co-culturing cancer cells with fibroblasts, immune cells, and others together to generate a situation that are closer to the real tumor microenvironment. Actually, I did not mention one of the paper we published last year in collaboration with University of Washington at Seattle. And we're actually using microfluidic chips to culture the tumor, the primary tumor itself. We collect the primary tumor after surgery and cut them into small pieces, into the micrometer size of structure, and then culture them in the microfluidic chips, and then look at their behavior or the response towards different chemotherapy drugs, and the correlation is quite similar. Yeah. Mm-hmm. So yes, there are a lot of research going on in this area.
Thank you.
Okay, thanks, Zhifang.
Thank you, Chufang. There's a hand up, Jilin Zhang.
Okay.
Hi,
Thank you
Jilin.
very much for the fantastic talk. So I actually have two questions. So in the last section, you mentioned the spatial transcriptomics in cancer, in the liver cancer. So what's your speculation about the relationship between the circulating tumor cells with the phenomena you observed, the difference of heterogeneity in the center and the front of the tumor samples?
Yeah, that's a great question, Ziling. We have yet to compare the transcription profiles of CTC versus those of the cells at the tumor margin or frontier versus those in the center, in the middle of the tumor. I suspect that the transcription profile of CTC will be much closer to those of at the frontier, because it's expected that CTC are generated from those frontier cells. But they will also have some difference because after they are subject to a number of different stress going into the circulation system, they will have some changes also continuously. But that's a very good question, and this is what we plan to do next, so that we have a more complete picture.
My second question is, if given you enough sample size, would you think you would be able to to build a model out of the data, like using cancer, using liver cancers, let's say, to build a model to simulate the migration of the CTCs and see its association with the heterogeneity. So, do you think whether it will work?
Yeah, that would be ideal, and that would be fantastic if we have sufficient sample and sufficient resources to carry out. For example, for each tumor, if we can perform the specially resolved transcriptomics for every section, and then we can reconstruct, similar like a confocal microscope, to reconstruct a 3D image of a cell. We can construct a three-dimensional tumor model with the transcription program at every position. And that would provide us with a very good, if not complete, understanding of what happened to every individual tumors at different stages and different locations. And this will also give us insight into how the cells evolve and develop and become metastatic. And so certainly, I mean, this will be the ambitious, but I believe this will be the direction that the cancer research are moving towards. And yes, to put it that way, yes, I think given sufficient sample and resources, this certainly should be done.
Yeah, thank you so much.
Mm-hmm. Thank you, Jilin.
Ying Li, please. You need to unmute yourself. You need to unmute yourself. We cannot hear you. You need to unmute yourself.
All right, so good.
Good.
Yes.
Good. We can hear you.
All right, great, Michael. This is my third time listening to your talk. There's a lot of new information for us to learning. In particular, we are in vivo systemic neuroscientists. We like to hear that. I did not really have a question. I have two comments. The first is I would like to add some area in the cancer metastasis is tumor neurointeraction. Because the tumor neurointeraction can promote cancer cells. Metastasis has been well recognized for a couple of years. Let's say peripheral nerve. In my early career in Michigan, I remember one of my colleagues, he identified the glial cell, which is the Schwann cells, can promote the breast cancer metabolism in the brain by immunocytochemistry, some early technique. I was involved at the time. I was just an assistant doing some surgery, but this is my impression. In the very first time, peripheral nerve, in particular glia, which is Schwann cells, involve cancer cell metabolism. And the recent couple of years in City University, we are involved in the brain. We are not intentionally to look at the extraglial or the dendrocyte glia related to cancer metastasis. However, during our learning, the background information is pretty strong. For example, we interested in extracyte really selected recently, as you know. And when is metabolic signal, metabolic energy supply, the one hand is signaling molecular. And we interested in relate to the learning memory, cognitive function. However, actually, the reactive gliosis was strongly around tumor metastasis cells. So it tells us the extracyte provide energy supply or signal molecule is not only related to physiological function for our neuroscience,
I don't know, Michael,
related
whether you
to
have a
molecular cell metastasis.
Okay.
Yes, thank you, Ying, for the comment.
The
Yeah.
other is the oligodendrocyte we interest into proliferation, differentiation, and malinformation. It's also related to metabolic cell proliferation by 5-fold increases. So that information tells me tumor glia introduction program is one way in your interest. I'm done, sorry.
Thank you, Ying, for the comment. Yeah, I make a quick comment. I think I'm not an expert in the brain related, but I think there are 3 aspects like in brain and cancer, glioma, of course, and also the brain metastasis of other tumors. And recently, actually, there's some study of how actually nerves are grow inside the tumor, in a solid tumor. And so there are some, they are nerves in the tumor. And this can generate from neural cells, neural stem cells somewhere. So this is a very early stage of research, and there's a lot of rooms to involve. Yeah. Thank you.
Okay, thank you very much. I don't see another question online or a raised hand. So I think we really witnessed a wonderful seminar. Again, you know, the sheer amount of information you provide and meaningful biological questions that you answer with your exciting technology is really quite amazing and quite convincing. With that, I really thank you, Michael, for this wonderful talk, this seminar. I thank you all for listening and for discussing, and can wish you a healthy weekend and beyond, and stay strong and stay tough, and see you next time. Thank you.
Thank you, Klaus, and thank you, everybody. Yeah, stay safe. Look forward to meeting you in person soon. Yeah, thank you.
Thank you.